Well, welcome. Really lovely to see you all. I'm very happy to see that it's such a popular session today. We've got almost 30 people joining us already at this moment. So really great to see all of you, and a really warm welcome to all the visitors and all the speakers. So this is part of the Early Career Research Seminar series by the Cambridge SRI in Reproduction. And we want to start off by saying we're very helpful to all the volunteer coordinators who've put this together in organizing this series, especially Daphne, Tara, Grace, Jane, and Ellen. And we're also very grateful to Christina Rossi, who's always doing a great job in getting everything going in the SRI, organizing everything. So thank you, Christina. And we're, of course, also very grateful to our two speakers today, Stacey Weiss and Amanda Rodgers, who will be giving short presentations. The seminars are held every second Thursday of the month, so this will be repeated at 1 p.m. And you can find details of the upcoming seminars on the SRI website, and Christina will post a link on that in the chat so you can stay updated about future events. So the Cambridge Reproduction SRI is an interdisciplinary initiative that brings together researchers across the University of Cambridge that all have an interest in reproduction. So it's everything from science, technology, and medicine to arts and humanities. Membership is open to all staff and postgraduate students and gets you access to SRI events, funding schemes. We currently have 4 different funding schemes for people at all different stages of their career. So it's a really good idea to get signed up, stay updated, because it might give you the opportunity to organize an event or develop your research. We have monthly newsletters, which has research highlights, events, and opportunities in reproduction research. And to join, all you have to do is download the membership form from the website, which Christina will post in the chat very shortly as well. And then you can send that membership form to Christina and you will become a member. And now we're going on to our second presentation, Amanda Rodgers. Amanda is a PhD student in the Department of Physiology here at Cambridge. And she will be giving a presentation today on the role of placental endocrine malfunction in the programming of disease in offspring.
Well, hi, I'm Amanda. I'm a PhD student in the Swartzi Perry Lab, and I'm currently about the halfway point of my PhD, so I'm just going to talk to you today about a little bit of what I've found so far in my project. So my project is based around the developmental origins of health and disease hypothesis, or what's often called the Barca hypothesis, after the epidemiologist David Barca. And this theory essentially states that adverse maternal environments, such as maternal obesity or poor nutrition, can lead to early life conditioning on the fetus. And this can lead to programmed changes in the offspring, which are maintained throughout life and can lead to metabolic diseases in adulthood. It's thought that this occurs in utero through programming changes to the cells, such as epigenetic changes, such as histone modifications or DNA methylation. And this theory also states that things that are referred to as a second hit, such as a poor diet, can also lead to exacerbation of these diseases in adults. So it can lead to worse outcomes in the adults. So since Barca first proposed this hypothesis, there have been multiple animal and epidemiological studies that have shown that adverse maternal environments can program the offspring's long-term health. But for the purposes of this talk, I'm just going to be focusing on insulin resistance and the programming of adipose biology. So insulin resistance and diabetes were among the first diseases that Barca and colleagues found to be associated with a poor maternal diet, poor maternal environment. And so low birth weight, which is often an indicator of poor maternal diet, environment and is linked with poor maternal nutrition, smoking, stress. And they found this is consistently linked to insulin resistance in the adults. And so there have been multiple maternal manipulation studies to try and understand the mechanisms behind this. And multiple manipulations have been shown to program insulin resistance in the offspring. However, really interestingly, they've found that different maternal manipulations have shown to program insulin resistance in different ways in different organs. So for example, diabetes has been shown to be programmed by decreased beta cell function or increased apoptosis of the beta cells in the pancreas in response to gestational diabetes or exposure to nicotine during gestation. And insulin resistance has also been shown to be programmed in the peripheral tissues, such as the liver, the skeletal muscle, or the adipose tissue by alterations in the abundance of the insulin signaling proteins in these tissues in response to various maternal manipulations. Thus, these studies in various animal models support the hypothesis that abnormal in utero environment can induce permanent changes in glucose and insulin's handling after birth and increase the risk of type 2 diabetes in adulthood. But key information on how these maternal manipulations are transferred to the growing fetus and the mechanism behind this remain unclear. So the developmental programming of adipose tissue in humans is usually looked at through the lens of maternal obesity, but there have been some really compelling studies in this area, such as studies done on mothers who have had bariatric surgery, and they've shown that the siblings who were born post-surgery when the mother was no longer obese had this decrease in adiposity compared to the siblings who were born pre-surgery when the mother was still obese. And there've also been some really interesting mesenchymal stem cell studies in this area where they've taken mesenchymal stem cells from the umbilical cords of infants, and they've found that these cells have an increased adipogenesis, and which correlated strongly to adiposity in the infants at 5 months when these infants were born to obese mothers compared to normal weight mothers. And so in animal studies, even though adipose tissue isn't often the focus of many DOHAD studies, alterations in adiposity and adipose biology have been found in many maternal manipulation studies. So maternal manipulations such as low protein diets, high fat, high sugar diets, or caloric restriction have all been shown to alter the adipose tissue. So you find things like increased hypertrophy in the tissue, increased hyperplasia in the tissue, or changes in browning in the tissue. But interestingly, in the adipose tissue, unlike the programming of insulin resistance, it seems that alterations in the adipose tissue are really dependent on the timing of the manipulation. And there's been some wonderful studies into this by Simmons and colleagues. And because adipose tissue is a highly sexually dimorphic organ, offspring sex seems to be play a real vital role. However, again, like the insulin resistance studies, the key information on how these maternal manipulations are transferred to the growing fetus and the mechanisms behind this remain unclear. And most importantly for us, the precise role of the placenta in the developmental programming of offspring is really unknown. So the placenta is the main interface between the mother and fetus, and it has a number of important roles throughout pregnancy. It regulates fetal growth. It's important for oxygen and nutrient transport to the fetus. And it also produces a number of hormones which regulate pregnancy. However, the role of placental endocrine function in the developmental programming is not well understood. So to investigate this, we use IGF-2 in our lab. And so IGF-2 is insulin-like factor 2, and it's a paternally expressed and imprinted growth promoting gene. And in humans, imprinting defects in IGF-2 have been associated with major fetal growth syndromes such as Beckwith-Wiedemann syndrome and Silver-Russell syndrome. And dysregulated IGF-2 is seen in the placenta in many complicated pregnancies. In the mice, very importantly for our work, IGF-2 has been shown to promote placental endocrine cell formation in vitro. And IGF-2 null mice have decreased fetal placental growth and decreased placental endocrine numbers. And some really interesting work that's been done by Beth in our lab, a different PhD student, has shown that IGF-2 knockouts in the endocrine cells of the placenta affects the hormone production. So in our lab, we utilize the mouse placenta as a model, and we do this because the mouse has structurally discrete placental zones. So the junctional zone, which is responsible for the endocrine function of the placenta, is structurally discrete to the labyrinth zone, which is responsible for the transport function of the placenta. And so by utilizing the mouse model, placental endocrine malfunction can be selectively induced. And we do this in our lab by utilizing the TPBPA gene, which is only expressed in the endocrine cells of the placenta. And so this is the model that I work on. It's the JZ-IGF-2 underexpression model. And so in this model, IGF-2 flocks males are mated to DPBPA-Cre females, and this produces offspring with IGF-2 underexpression only in the junctional zone of the placenta. And so it's a whole litter manipulation, but IGF-2 is unaltered in the placental transport zone, in the offspring, and in the mothers. And for this study, litters of the reverse parental cross were used as controls. So the in vivo work for this study was done before I arrived at the lab, and it was carried out by two postdocs in our lab, Hannah Young and Jorge Lopez-Tello. And what they found was that male and female fetuses were significantly smaller than controls. However, by 3 weeks of age, the offspring of both sexes showed normal weight. And when they performed insulin tolerance tests on these animals at 12 weeks of age, they found that these animals were significantly insulin resistant. And they also performed DEXA scanning on these animals at 13 weeks of age. And what they found was that in the females, in female JZ-IGF-2 animals, they had a decrease in percentage adiposity, whereas in the males, there was no change in percentage adiposity. So this is where I arrived to the lab. And so my aims were to determine whether the programmed alterations in offspring metabolic profile in response to JZ-IGF-2 underexpression are associated with changes in insulin production and insulin signaling in key metabolic organs, and also lipid handling, morphology, and the biology of the white adipose tissue. So for aim 1, I looked into the key insulin metabolic organs, such as the pancreas, which is responsible for producing insulin, and also into the liver, skeletal muscle, and adipose tissue, the main insulin responsive organs. So briefly, this is the experimental design of the in vivo work. So litters of the IGF-2 underexpression and the reverse cross were standardized at day 3 to 3 male and 3 females. Then at week 3, these were weaned onto either a chow or a high-fat, high-sugar diet. And at week 12, metabolic tests were performed, such as glucose tolerance tests and insulin tolerance tests. And then at week 13, these animals were DEXA scanned and the tissue was collected. For today's presentation, I'm just going to be talking through the chow data, but I'm more than happy to talk about the high-fat diet group at the end if anybody wants to ask questions on them. So the first thing I did was to look at the pancreatic insulin content to see if the insulin resistance that we were seeing was being caused by a decrease in pancreatic insulin production. And actually what we found was that in the males, there was no change in insulin content in the pancreas. However, in the females, we saw this increase in pancreatic insulin content. And we thought this was interesting because this is sometimes observed in pre-diabetic animals, where the peripheral tissues have become insulin resistant. So the pancreas compensates by trying to produce more insulin. So we then started looking at the peripheral tissues. And to do this, we looked into insulin signaling pathway proteins. And so when insulin binds to its receptor, it dimerizes and autophosphorylates and causes sort of a cascade of reactions that happen in the cell. So it interacts with multiple pathways. So it activates the MAPK pathway, which is responsible for cell proliferation and survival. It also activates AKT, which has downstream effects on glycogen synthesis, and also interacts with mTOR1, which is a key hub in the cell, and is responsible for things like protein synthesis. So it has multiple functions in the cell. So we chose a few key proteins to have a look at. And so first we looked at insulin signaling proteins in the liver, and we did this via Western blot. And so you can see in the females, there was this decrease in IGF-1R and a decrease in P110 alpha. And in the males, we saw this decrease in AKT and a decrease in phosphorylation of 4-EBP and a decrease in phosphorylation of JSK3. However, the pathways weren't consistently downregulated, though we do see consistent differences between the JZIGF2 underexpression animals and the control animals, nothing that necessarily explains the insulin-resistant phenotype that we were seeing. So we wanted to see if these changes led to compositional changes in the liver. And in the males, we saw this increase in protein levels of JSK3 beta, but decrease in phosphorylation of JSK3 beta. And these seem to cancel each other out and lead to no change in glycogen content in the liver. Whereas in the females, we saw this alteration in phosphorylation of JSK3 beta and this alteration in glycogen content in these animals. And so we moved on to lipid handling in the livers, and we saw this increase in lipoprotein lipase in the females, which seemed to correlate to an increase in lipid content in the livers of these animals. Whereas in the males, we saw this, again, an increase in lipoprotein lipase, but this decrease in fatty acid synthase, which seemed to, again, cancel each other out to no change in lipid content in these animals. So then we looked into the insulin signaling pathway in skeletal muscle. And though we do see a few changes between the JZIGF2 underexpression animals and the control animals, there was nothing that was clearly, that was explaining the insulin resistant phenotype that we were seeing. And again, we looked into lipid handling in the skeletal muscle, and nothing really that explained our phenotype. So finally, we looked into insulin signaling proteins in the gonadal tissue, and we did see this decrease in MAPK and this decrease in JSK3 beta. However, again, there were no changes that clearly explained the phenotype that we were seeing of insulin resistance. And we did also look into lipid handling proteins in the gonadal tissue, and we saw no changes. And we also looked at these through qPCRs, and the qPCRs were consistent with the results that we were seeing in the Western blots, but again, nothing that changed our, that explained our insulin resistant phenotype. So to summarize AIM2, the insulin signaling pathways were affected in all three metabolic organs, and these changes were sexually dimorphic. However, these changes did not fully account for the programmed insulin resistant phenotype or changes in the adiposity in response to JZIGF2. So then to move on to AIM2, we looked at lipid handling, morphology and biology of the white adipose tissue. And we did this because if you can remember from the DEXA scanning data that I presented at the start, that in the females, we saw this decreased percentage adiposity in the JZIGF2 underexpression group, whereas in the males, there was no change in adiposity. So we wanted to have a closer look at the adipose tissue. And when we looked at the morphology of the gonadal adipose tissue, we found this significant decrease in adipocyte cell size in the female JZIGF2 underexpression group compared to the female controls. And in the males, we didn't see any change in adipocyte size. And so this led us to thinking, well, what affects adipocyte morphology? And while adipose tissue is an endocrine organ vital for whole body homeostasis and metabolism, and as such, it's a highly plastic tissue with multiple ways of affecting adipocyte size. The tissue can be hypertrophic, in which case the cell size is increased. The tissue can be hyperplasic, in which case the cell size of the adipocytes is decreased, but they have an increase in cell number. And there are also different adipocyte types. So you can have white adipocytes, which are the type that's typically found in the gonadal tissue. And these are quite large. adipocytes with one lipid droplet, and they're around 100 micrometer in diameter. You can also get brown adipocytes, which are the type of adipocytes that are typically found in the interscapular region. And these are much smaller adipocytes, so roughly about 20 micrometer in diameter, and they have multiple lipid droplets. And these two cells derive from different cell lineages. However, you can also get these what's often called bright or beige adipocytes, which are phenotypically similar to the brown adipocytes. However, they evolve from the same cell lineages as white adipocytes. So we then looked into the expression of key genes in the gonadal adipose tissue, and we did this through qPCR. And we looked into markers for hypertrophy, hyperplasia, lipogenesis, and lipolysis. And we didn't see any changes between the control and the JZIGF2 animals in either the female or the males. So by process of elimination, we started looking into browning markers. And much to our surprise, we started to see this the increase in browning markers in the female JZIGF2 underexpression animals in the females compared to the controls, and no change in the males. So we wanted to test this. So fortunately, we also had the retroperitoneal fat tissue that was taken from these animals. So we looked at the browning markers in the retroperitoneal fat tissue. And again, we saw this upregulation of brown markers in this tissue. So we wanted to see if these gene changes translated to changes in the protein expression. So we looked at UCP1 protein expression in the gonadal fat tissue, and we did this through Western blot. And again, we saw this increase in UCP1 in the females, and this no change in the males. And we also checked this through immunohistochemistry. So to summarize, aim 2 is that the decrease in adiposity in the female JZIGF2 was accompanied by this decrease in adipocyte cell size and this increase in browning adipocyte markers. And these changes were not observed in the male JZIGF2 underexpression offspring. So to conclude, placental endocrine function is important in determining the metabolic health outcomes of the adult offspring, and this is mediated by programmed changes in insulin production and insulin signaling and adipose tissue biology. And so as I'm at the halfway point of my PhD, I've currently got to the stage where I have generated more questions than answers. And so I'll just talk you briefly through my current work. And so my first of the current work is I'm trying to determine the complete repertoire of molecular changes occurring in the adult offspring adipose tissue in response to JZIGF2 underexpression. I've done this by sequencing the gonadal fat tissue, and this is currently being analyzed. I'm also trying to examine whether changes in the adipose biology and insulin signaling are present in the JZIGF2 offspring during earlier postnatal life. So I'm looking into doing some mesenchymal experiments and doing insulin tolerance tests, metabolic age assessments, TDN-MR scanning, and molecular analysis of tissue from animals of younger ages. And then finally, I'm trying to explore whether the changes in offspring metabolic phenotype are caused directly by the altered endocrine output of the JZIGF2 underexpression placentas. So for this, I'm doing some conditioned media experiments utilizing a technique that was recently developed in our lab by a postdoc, Tina Naxo. And so finally, I'd just like to say thank you to my supervisor, Dr. Susie Perry. I'd like to say a huge thank you to Miguel and Ianel, who developed the animal model, and a huge thank you to Hannah, Jorge, and Effie, who worked really hard to get all the in vivo data. And then a huge thank you to all past and present members of the Sfrutzi Perry Lab for all their advice and input and help all the time. Thank you for listening.